In the last lecture, we discussed carbohydrates and naming carbohydrates and other basic things about carbohydrates. In this lecture, we're going to discuss the properties of carbohydrates and reactions that can be done with carbohydrates. So let's get started. The first one is an oxidation reaction. So, as we discussed in the last lecture, very small amounts of the open chain are actually present in solutions. And when they're in the open chain, we have an aldehyde here that's sitting right next to hydroxyl. And in the presence of an oxidizing agent, such as Medic solution, which has copper in it, copper 2 to be exact, we can oxidize the aldehyde to a carboxylic acid. And in that process, the copper 2 actually becomes a copper 1 because it was reduced. When that happens, we rename glucose, which is in this example, to gluconic acid. And it goes without saying that both of these are the D versions because in nature, sugars are all D versions, right? So when a carbohydrate is capable of doing any kind of reduction, so in this case, the glucose reduced the copper 2 to copper 1, we call that a reducing sugar. Make sure you know that. So we can also oxidize fructose to glucose as well, right? So fructose, remember, is a ketohexose, right? Which means it has a ketone. So here is fructose, and here's the ketone that is in question, right? Using basic Benedict's solution, you can actually rearrange this ketone into an aldehyde. And when you do that, the fructose becomes a glucose, right? No longer is it a ketose because the ketone is gone. It's now an aldose because glucose has an aldehyde right here, right? We can also reduce monosaccharides using a platinum catalyst and some hydrogen gas. So here we have D-glucose again, and when it gets reduced, it becomes D-glucitol or D-sorbitol. Funny story about sorbitol. Sorbitol is actually known as a sugar alcohol, and many of the sugar substitutes that we use in like low sugar or sugar-free foods are actually sugar alcohols. They're normal sugars like glucose that have been reduced so they become sugar alcohols. So some of those are actually listed here: sorbitol, xylitol, xylose, mannitol, and mannose. You see mannitol a lot in like gummy bears and stuff like that. Speaking of gummy bears, this is actually a nutrition facts label from Haribo's sugar-free gummy bears, right? And if you take a close look at it, sorbitol is actually one of the listed ingredients in sugar-free gummy bears. And so the reason why it's a funny story is because back when I was in grad school, I had an undergraduate working in my lab, and we actually use sorbitol to help in the expression of proteins and enzymes when we grow cells. And so there was this big bucket of sorbitol. I needed some of it, so I asked an undergrad of mine to actually pull the bucket down from the top stock so that we could get the sorbitol. Well, he pulled it down, but in the process, he spilled some of it on himself, right? And some of it may have fallen in his mouth. And so we went and looked for the MSDS sheet for the sorbitol to make sure that he wasn't ingesting anything that was toxic. And so the funniest thing happened when you type in sorbitol into Google. It comes up with all these Amazon reviews for Haribo's sugar-free gummy bears. And these reviews are quite hilarious. So if you've never read them before, I encourage you to do that. Just type in Amazon reviews, sugarless gummy bears, or something to that effect, and you'll find them. They're quite funny. But So to end the story, we we asked him the next day if he had any gastrointestinal issues because of the sorbitol, and he said no. So we're pretty sure he didn't ingest any of the sorbitol. But that being said, I forgot to mention that when a sugar that ends in cose, like glucose, gets reduced into a sugar alcohol, the name changes to etol, right? So that's why it's D-glucose has turned into D-glucotol, or in this case, sorbitol, D-sorbitol. Sorbitol is easier to say. So let's practice a reduction of a monosaccharide. Pretty simple, right? We have D-mannose here, and we're going to turn it into D-mannitol. We're going to need some hydrogen gas, and we're going to need some platinum catalyst to do a reduction here, right? And when we reduce an aldehyde, which is this guy right here, right? It becomes a sugar alcohol. So let's draw out the open chain. H, H, OH, OH, H, H, OH, OH, right? And then down here it stays the same because there's nothing to reduce down here. C2, CH2OH, and the aldehyde becomes CH2OH as well, right? We gained 2 hydrogens in here, whereas there used to be only 1 hydrogen that came from this hydrogen gas, right? And that is our sugar alcohol, mannitol. I believe there's 2 N's in mannitol. There we go. Alcohol's on the right side, so it's a D-mannitol. Don't forget that. So as far as applications go, when it comes to the oxidation reduction chemistry of monosaccharides, we can do glucose testing. So we all know that the kidneys absorb glucose from the bloodstream, right? But if your blood sugar exceeds 160 milligrams of glucose per deciliter, the kidneys can't handle that. And so what happens? That sugar will spill over into the urine, right? And this is a medical condition known as glucosuria. There's actually a test that we use that involves the same kind of chemistry, oxidation reduction chemistry, right? That can help us determine whether or not a person is suffering from this condition, right? So here's glucose in the bloodstream, D-glucose in the bloodstream, right? In the presence of oxygen and glucose oxidase is what is glucose oxidase? It's an enzyme that does oxidation, right? It can actually oxidize the aldehyde of glucose to become a carboxylic acid, right? And when that happens, glucose becomes gluconic acid. But that's not the part that you can use to diagnose somebody with glucosuria. One of the byproducts of this reaction is hydrogen peroxide. And hydrogen peroxide, when you add it to a dye known as 2-methylaniline, with the peroxidase enzyme, you get a colored solution, right? You get a colored solution. And the color of the solution actually indicates the concentration of glucose in the urine. So here's blue, and blue is 0, right? And remember, we don't want it to exceed 160, right? So we're looking when we're doing this test to find find test results that are in between blue-green, which is 100 milligrams per deciliter, and green, which is 250. Anything bigger than that, you have a problem, right? So anything bigger than green to dark brown is bad. How does this work? Well, it works because for every molecule of gluconic acid that is produced from this reaction, there's also a molecule of hydrogen peroxide. That means that the concentration of gluconic acid is the same as the concentration of hydrogen peroxide. And that's why you can use that as a way of detecting what the concentration is of glucose in the system. So that's pretty interesting. And this is actually a photo of the actual test right here. The color test. The package comes with little colors on it, and you compare the color of the test strip to the colors on the side of the bottle. So if you've ever done any testing with like pool water or with fish tanks, they all come with the same kind of test strips where you dip it into the water and then it comes out and it changes colors. And when it changes colors, you just compare that to the key, which is either on the bottle or in the instruction manual. Pretty interesting stuff. So enough about monosaccharides. Let's talk about disaccharides now, right? So what's a disaccharide again? It is two monosaccharides that are linked together. When you have two glucoses linked together, you have maltose. When you have one glucose and one galactose linked together, you have lactose. And when you have a glucose and a fructose together, you have sucrose, right? So the most common disaccharide sugars that we encounter are maltoses, lactose, and sucrose. Let's first talk about maltose, known as malt sugar. It's interesting because it is two alpha D-glucose molecules that are joined together by an alpha 1,4 glycosidic bond. It's found in cereals, candies, and brewing, and it can be obtained through the hydrolysis or hydrolysis, using water to cut molecules apart, of starch. What's starch? Starch is just a polysaccharide. Many of these glucose molecules attach to each other, which we'll talk about a lot later. But the important thing to notice, though, the important thing to remember, though, is that maltose is those two glucoses linked through an alpha 1,4 glycosidic bond. What does that mean? It means that it's an alpha because the oxygen that forms this bond, right, came from this first glucose, and that oxygen, that hydroxyl that it came from, is pointed down, right? So it's an alpha glucose, and that's what forms the alpha in the 1,4 glycosidic bond. It's 1 because it's on the first carbon of that first glucose, and 4 because it's on the fourth carbon of the second glucose. So remember that maltose has a alpha 1,4 glycosidic bond. It'll come back to you later. Then we have lactose, which is milk sugar. That's a disaccharide as well, of course. It's basically found in dairy products like milk or cheese. It's about 6 to 8% of human milk and about 4 to 5% of cow's milk. The interesting thing, though, is that it's composed of a beta D-galactose and an alpha or beta D-glucose. And its linkage, as you can see here, is pointed upwards, so it forms a beta 1,4 glycosidic bond, right? In contrast to the maltose, which had the alpha 1,4 glycosidic bond, right? Lactose has a beta 1,4, and that's important for something else later. Something else later. So keep that in mind. Put a pin in that. The beta 1,4 glycosidic bond of lactose. Last but not least, we have sucrose, which is the table sugar. Of course, it comes from sugar cane or sugar beets, and is composed of an alpha D-glucose and a beta D-fructose. The interesting thing about sucrose, though, is that its linkage can be either an alpha or beta 1,2 glycosidic bond, right? Not like the others, which have a 1,4. Why is that? Well, the first reason is because fructose is a 5-membered ring, whereas the other two were linked to 6-membered rings, right? And so the 1 comes from the very first glucose that's in the disaccharide from carbon 1, and it's linked to carbon 2 of the fructose. And that's why we have a 1,2 glycosidic bond. And it can be either alpha or beta. The thing about sucrose, though, is that it can't form an open chain, it cannot be oxidized, and Benedict's reagent, it cannot react with Benedict's reagent, and it cannot reduce a copper, like what happens when you use Benedict's reagent. So let's talk about the sweetness of sweeteners now. So all the sugars and the artificial sweeteners, they differ in sweetness, but everything is compared to sucrose, how sweet something is, right? And it's compared to the fact that sucrose or table sugar has a sweetness of 100, okay? So things like galactose the monosaccharides have a sweetness of 30 or 75, right? Compared to sucrose, which has 100 again. But fructose by itself is actually even more sweet than its combination of fructose and glucose together, which is sucrose, right? So that's interesting. It's interesting that fructose, which is a part of sucrose, is sweeter than sucrose. Compared to the other disaccharides, lactose is at 16, maltose is at 33. So both of those are actually less sweet than sugar, than sucrose, table sugar. But then we get into the sugar alcohols, like sorbitol in our gummy bears. It's only at a 60. Only 60% of the sweetness of sugar. Mannitol, 80. Xylitol actually has the same sweetness as table sugar. But then when we go into the non-carbohydrate sweeteners, right? Sweeteners that are not derived from carbohydrates, like sucrose or any other carbohydrate. Things like sativa, aspartame, saccharin, right? Those are things like Equal and Splenda and stuff like that. Sucralose, neotame, and advantame. My God, they have orders of magnitude more sweetness than sucrose does. So you don't have to use as much. So what are these artificial sweeteners? Well, like I was saying before, there's aspartame. And aspartame is commonly known as NutraSweet or Equal, right? And its shape, its molecular structure is there. The reason why it's considered a non-carbohydrate sweetener is because notice that it's not made of carbohydrates. This is actually 2 amino acids put together. The aspartate amino acid and the phenylalanine amino acid, which you'll learn a lot about later in the semester. But the point is, they're amino acids. They're not carbohydrates. Sucralose, though, that is a derivative of a sugar. It's marketed as Splenda, and it actually comes from sucrose. And the difference between it and sucrose is that sucralose has these 2 chlorines on there instead of hydroxyls. So this actually happens a lot, where chemists will go through and change substituents of known molecules that have a certain effect, hoping to increase the effect or make it more efficient by changing some of the constituent molecules, constituent atoms, but retaining the original shape, the original structure. So that's pretty interesting there. So saccharides play a huge part in blood type as well. Those carbohydrates are what determine whether a person has type A blood, type B blood, type AB blood, or type O blood. So when you have type O blood, your red blood cells on the surface have these three saccharides: an N-acetylglucosamine, a galactose, and a fructose. And that's it. Right? The other two blood types, A and B, have those original three plus one more. For type A, there's the extra acetyl N-acetylgalactocosamine. And then for type B, there's an extra galactose. And then when you have type AB blood, you have both of these in your blood. Right? So that's the reason why type O blood is the universal donor. Because when you introduce O blood to a type A, type B, or a type AB, their system is already familiar with having those three original N-acetylglucosamine, galactose, and fructose saccharides attached to the red blood cell's surface. The problem comes when you try to introduce one of these extra saccharides into a blood type that doesn't have them. For example, trying to donate type A blood into O-type blood patient, right? That would create an immune response because this would be considered an antigen, right? It's something very foreign to that body because that body is used to having red blood cells that only have those three saccharides, not four saccharides. And that's why you can't donate type A blood to a type B patient. Type O patient. But you also can't donate it to a type B patient either. Because the type B patient is used to having the extra galactose in that position, not the N-acetylgalactocosamine, right? So when you have type AB blood, you actually have both of these, right? So you can receive any blood because your body is used to having just the three original and used to having the three original plus acetylgalactocosamine, or the three original plus the extra galactose. And that's why type A blood type is the universal recipient. So polysaccharides are most commonly used to store glucose. And in plants like potatoes, that comes in the form of starch. Well, starch is made up about 20% of a polysaccharide known as amylose. That consists of about 250 to 4,000 alpha-D-glucose molecules that are all connected together with an alpha-1,4 glycosidic bond, right? And even though this is a straight-chain polymer, the polymer tends to kind of coil itself into a helical structure. But the thing to remember is that those glucose molecules, or monomers, are joined together by this alpha-1,4 glycosidic bond. Well, if 20% of starch is amylose, what's the other 80%? If 20% of starch is amylose, then what is the other 80%? Well, it's called amylopectin. Amylopectin is actually very similar to amylose, except that it's a branch chain polysaccharide, right? Where the glucose monomers can be attached by either an alpha-1,4 or an alpha-1,6 glycosidic bond, right? The alpha-1,4 is when it's connecting via straight chain, And alpha 1,6 glycosidic bonds form when it branches off from the main chain. And you can see that here, where we have the straight chain coiling up of the amylopectin, but over here we have a branch come off, right? And that's where a 1,6 alpha 1,6 glycosidic bond would be, right? And it keeps on coiling, and you see another branch over here. And it coils again, and then just branches off. And all those branches begin with a alpha 1,6 glycosidic bond, right? Why is it 1,6? Because the branch happens at the 6th carbon of the glucose molecule. And then the 1,4, again, because carbon 1 of the first glucose and carbon 4 of the second glucose. And the alpha because the oxygen is pointing down. Remember that. That's important. These starches hydrolyze easily in water and acid and give smaller saccharides known as dextrins, right? Then they hydrolyze even further into maltose, which is a disaccharide of 2 glucoses, and then finally to glucose, which is what happens in your body when your body starts to metabolize glucose for energy, right? In animal cells, though, glucose storage is actually found in the form of glycogen. This polysaccharide, right, is very much like the other polysaccharides, amylopectin and amylose, right? But it's much more highly branched. Okay, much more highly branched. These are also made with the alpha 1,4 glycosidic bonds for the straight chain part. And then the branches are accomplished at the 1,6 glycosidic bonds at every 10 to 15 glucose units. So the branches happen way more frequently. The picture you see here at the right is actually a picture of a huge glycogen molecule. But glycogen just doesn't start out on its own. It actually has a small protein that starts it in the center, known as glycogenin, right? Glycogenin. And glycogenin actually is the initiator for forming glycogen molecules. So the interesting thing about that is glycogen is actually stored in both our livers and our muscles, right? And the muscles actually store about 24 hours worth of glycogen that you can use to use as fuel. But our liver stores about 24 hours of glycogen that can be used as fuel. Not the glycogen itself, but it will break down into glucose. But again, important to note, alpha 1,4 glycosidic bond, alpha 1,6 glycosidic bond. And those are the storage molecules for glucose in both plants and animals. We have one more polysaccharide to talk about, which is cellulose. Cellulose is also made of glucose units, right? Except that they're linked together with a beta 1,4 glycosidic bond, right? Not an alpha. Now, plants don't use cellulose for storage. It's actually structural. It's actually made for the cell walls of wood fiber and plant cells and such like that. It is much more resistant to hydrolysis than the starches, which makes sense because when you're storing sugars for energy later on in life, you want it to be easily hydrolyzed so you can break the sugars down and use it to make ATP. But if you're using those carbohydrates or those polysaccharides as structure, you want them to be more resistant to hydrolysis because you don't want it just to break apart when it's a part of your structure, right? So this is important because humans cannot break down the beta 1,4 glycosidic bonds, which are the plant cells, right? So let's just review something real quick, right? All the other glycosidic bonds that we were talking about. If you recall, maltose has an alpha 1,4 glycosidic bond. Lactose, well, we'll skip lactose. Sucrose has either an alpha or a beta 1,2 glycosidic bond. Amylose has an alpha 1,4 glycosidic bond. Amylopectin has an alpha 1,4 and an alpha 1,6 glycosidic bond. And glycogen has an alpha 1,4 glycosidic bond. All of those are either alpha 1,4 glycosidic bonds or they're 1,2 glycosidic bonds that have the beta. Only one is like cellulose, which is lactose, that has a beta 1,4 glycosidic bond, right? So it turns out that the same way that humans cannot break down a beta 1,4 glycosidic bond because it's the cellular structure of plant cells, right? Some humans cannot break down lactose because of the beta 1,4 glycosidic bond. So that's the basis for lactose intolerance. Very interesting stuff.